welcome everybody to this webinar tonight. We're really appreciative of your attendance. It's going to be a great interactive webinar tonight with Dr. Linda Hiraki. She's going to be speaking about deciphering the genetic complexity of lupus, insights, and future avenues. She's spearheading a large-scale international cohort study of systemic lupus erythematosus patients, following them prospectively over years to understand the genetics of lupus, its diverse manifestations, disease progression, and associated outcomes. Additionally, she's at the forefront of research efforts to uncover rare genetic alterations linked to systemic inflammatory diseases, including studies focused on monogenic lupus and lupus-like disease. We are going to be exploring the intricacies of this genetic complexity and understanding what it entails for Dr. Hiraki and the patients participating in these groundbreaking studies. Dr. Hiraki is a clinician-scientist in the Division of Rheumatology and scientist in Genetics and Genome Biology Program at SickKids Research Institute. Dr. Hiraki completed her medical degree at Queen's University and her clinical training in paediatrics and rheumatology at SickKids. She is certified by the Royal College of Physicians and Surgeons of Canada in the specialties of pediatrics and pediatric rheumatology. Hiraki went on to pursue research training at the Harvard T.H. Chan School of Public Health, where she completed a master's and Doctor of Science in Epidemiology. She holds a Canada Research Chair in Genetics of Rare Systemic Inflammatory Diseases. So as you can see, we have a true expert here with us tonight. A couple of housekeeping things to let you know about is the chat is available, and there are going to be some interactive questions going on tonight. But since you cannot open your mic or have your screen on, you can answer in the chat. There's going to be a few multiple choice questions as we go, and I'll just call out what the answers are. Also, in the Q&A function, please put any questions in that you have tonight. We are going to have a Q&A period at the end of the talk. So I will be reading those questions out so Dr. Hiraki can answer them. So you can pop them in there at any point. We just want to ask you not to take any screenshots tonight. You may have heard us talking when we were just talking about the recording. We are recording this tonight, but there's a section of it we're not going to be able to show on our YouTube channel later. So for those tuning in YouTube on this, there's a section we will be marking out as upcoming news and research. My name is Lisa Bilodeau. I'm the Vice President of Lupus Ontario, and I'm honored to turn you over to Dr. Hiraki.
Thanks so much, Lisa, for that very generous introduction. And thanks to everyone joining tonight. I am very grateful that you're taking time out of your evening to be here. And as Lisa mentioned, I'm really hoping this is an interactive session. I have some questions for people to chime in on. And if I have gone off in some wild direction and you're like, wait a minute, I want to catch up with this train, please raise your hand. And Lisa will make sure that I pause because when I share my screen, I may not be able to see your hands and things. So with that, let's sort of kick it off now. It's going to be a little clunky for a minute. And here we go. How does that look, Lisa? Is that looking okay? Excellent. All right. So Lisa talked about this lovely title, which I can take no credit for. I think the very clever folks at Lupus Ontario suggested this, and I think it was great. So what I'm hoping to do is to start off by giving a little bit about genetics. I should mention my disclosures. I serve as a consultant for Janssen. We'll begin by talking about systemic lupus erythematosus, which I think most people on the webinar are very familiar with. It is a very clinically heterogeneous disease, also called the disease of 1,000 faces, because it looks different in everyone. And of course, it can change over time. It's a chronic disease. Unfortunately, to this day, we still do not have a cure. It's one of the quintessential autoimmune diseases where the immune system, which typically serves to protect people from infection, starts attacking one's own healthy tissues. And it is life-threatening. It's estimated that in Canada, the prevalence of lupus is about 50 and 100,000, and this number tends to vary a great deal depending on the population that's being studied, anywhere from 20 to 400 per 100,000. So I'm going to just kick it off with a question for everyone. What factors have been identified to increase the risk of developing lupus? A, genetics, B, infection, C, ultraviolet or UV exposure, D, smoking, E, all of the above. So if you'd like, feel free to drop your answer into the Q&A or the chat. I'm not going to look at who's saying what, so feel free to just throw out whatever you want.
We only have a couple answers so far saying E.
Okay.
Does anyone else want to weigh in on the chat? More E's?
Okay. Oh, you clever people. Yes. All of these things have been implicated as factors that can increase the risk of developing lupus. And I think, like with so many human traits, lupus is one of these things where we understand both environmental factors, like those ones I listed, infection, UV exposure, diet. There have been elements in each of those that have been identified as potentially contributing to lupus risk, as well as genetic factors or things that are heritable. And the reason why people think genetics is important began with looking at the epidemiology, or the phenomenon that we see in who is affected by lupus. So we know that there's a strong female predominance in lupus. In adults, the ratio of females to males is about 10-1, and in children, 4-1. There has also been observed a much higher concordance. So similar family members who are affected in monozygotic twins who are genetically identical versus dizygotic twins who have genetic sharing similar to siblings who are not identical twins. And what has been observed is there's a 10-fold increased risk of lupus in genetically identical twins versus fraternal twins who are like siblings. There's also variation across global populations when it comes to the prevalence. And we know that one of the major differences between different ancestral global populations is genetics. And lastly, when people have tried to estimate how much of the risk of lupus is due to environmental factors versus genetic factors, it's estimated to be about 66% due to genetics. So these are, I think, overall where we stand when it comes to our understanding of what plays an important role in developing lupus. So what I hope to cover in the webinar today is really beginning with the foundation of what are genetics? What are we talking about? What do we mean when we talk about common and rare genetic variants? The role of sex, the role of ancestry. I'll talk a little bit about our work looking at the genetics of specific lupus manifestations, in particular age at when a person's diagnosed with lupus. I'll talk a little bit about monogenic lupus. And if there's time, I hope to talk about the genetics of a serious complication of lupus called macrophage activation syndrome. Okay, so what are genetics? Really, we're talking about the study of heredity, or the expression of traits and phenotypes. And these are these things that are passed down from generation to generation. And when we think of the classic heritable diseases, you might think of them as two groups. There's Mendelian or monogenic traits, and some of the classic diseases are Huntington's disease, which is considered an autosomal dominant disease, where all it takes is one change in one of your chromosomes that can lead to disease. And this is a family pedigree where you're seeing father, a male, and a mother, a female, and their offspring listed here. And the ones that are in the darker boxes are affected family members. And you can contrast this to an autosomal recessive disease like sickle cell anemia, where someone might carry a variant, but it takes 2 copies before someone can actually manifest the disease itself. So here you see a father and a mother having offspring, and one of their children is affected, 2 are carriers, and 1 is unaffected. So this is sort of that classic monogenic heritable pattern. And then we can contrast that to polygenic traits. And a classic example is coronary artery disease, where we know that genetics plays an important role in who is at risk of developing coronary artery disease, but it is typically a disease where you have hundreds of changes across the genome, each of them contributing relatively small risks to developing coronary artery disease. So it's considered a polygenic trait, many, many different changes together. And when we're talking about these genetic changes, we have to go back to the foundation of this, the DNA, the deoxyribonucleic acids. And this is the double helix. A represents one chromosome, B would be another. And what we're looking at are these alleles. And what I'm highlighting here in the boxes is how on one strand, someone might have an A allele, and then on another, they might have a C allele. And these are called single nucleotide polymorphisms. And in this instance, let's say your risk allele, the allele that's associated with lupus per se, someone could have one copy of the C, they could have two copies, or they could have no copies. And so when we're talking about these changes, this is the sort of thing that we're talking about when people are talking about SNPs. And when you look across the genome, there are 3 billion base pairs that make up the genome. But only a small fraction of that, 1%, is the exome. This is the part of the genome that codes or is the instruction manual for creating proteins. And we know that in Mendelian disorders, the majority of the changes that we see are in these exonic regions, the exome, the one responsible for protein coding. And a lot of the changes we know from the non-coding regions are ones that are common and made up and discovered with genome-wide association studies. So why study genetics? I am very biased in this. I think studying genetics is incredibly important for a number of public health goals. As I was mentioning before, quantifying genetic risk can be really important for our understanding of the disease. Knowing about the genetics can improve prediction for individuals at risk. It can provide deeper insights into the specific biology of the disease, and that in turn can help us design and implement targeted biologic interventions. All of these things to the ultimate goal of helping us prevent and treat and ultimately cure diseases. So I'm revisiting this concept of heritability. This is a graph I created comparing different features, phenotypes, and diseases with regards to their heritability. So they're listed on the bottom, and then the heritability is on the y-axis. And so a heritability of 90%, which has been reported for type 1 diabetes, close to that for psoriatic arthritis. Height is also something considered highly heritable. This is where it's been found that a large proportion of how tall someone will get, for example, can be determined by their genetics. And if you look across this chart, you see that lupus is one of these traits that has a large degree of heritability. And when we think about the effects of each of these changes across the genome, it can be useful to think about how the prevalence or how common a variant is in the population tends to be related to its effect. So in this graph, which is rather old now, allele frequency or how common a genetic change is in the population is listed on the x-axis. So all the way at the left, you have things that are very, very rare. Less than 1% of people in the population will carry these changes. And part of the reason why it's thought that these things are rare is because through generation to generation, their effect is so severe, it reduces the likelihood that they're going to be passed on to the next generation. So these are usually changes that are having very large effects on the outcome that we might be interested in. All the way on the other side, you see these common variants that maybe are present in 5% or more of the population. And it's thought that generally these changes have modest or low effect individually. So the way we pick up these different changes also is different. Common variants tend to be picked up using arrays, And rare alleles tend to be picked up by sequencing, where you're individually characterizing nucleotides across the genome. That's called whole exome sequencing or whole genome sequencing. And I think as time has gone on and as the technologies have advanced, we're learning more and more about all of these variants and their relative effects on a range of human traits. So generally, systemic lupus erythematosus is considered a polygenic disease for most people, where we believe hundreds of changes across the genome are contributing to someone's risk of developing lupus. However, we also know that there is a subset of people who also have a monogenic form of disease, where a single change is likely responsible, or it has been demonstrated to be responsible for their lupus disease. Okay, so now this is the next question. We can see how effective I am at giving this talk. For most people with lupus, and I'd ask you to complete the sentence with the best answer. A, genetics are not risk factors. B, a change in one gene leads to lupus. C, changes in 10 genes increases the risk of lupus. Or D, changes in over 100 genes increases the risk of lupus.
Okay, we've got some answers. A couple of them say B. Someone is saying D. We have C. So we have a range.
A whole range.
Yeah.
Okay. All right. So maybe I didn't do a good enough job. The key in the lead up was for most people. So for most people, the answer is D. People are absolutely right. There are some people in whom a single genetic change we believe is responsible for disease, but for most people with lupus, it's been demonstrated that it's a polygenic disease where you have more than 100 changes in genes across the genome that are contributing to disease. Now, how do we know this? So there have been a number of genome-wide association studies that have been done to try to identify these changes. These are two Manhattan plots that have come from some of the largest genome-wide association studies of lupus. And a Manhattan plot was called a Manhattan plot because apparently this looks like the Manhattan skyline. It's clear scientists don't get out much. But what you have on the x-axis are each of the chromosomes in different colors. And on the y, you have the negative log 10 p-value. So this is a measure of significance. The higher a dot is, and each dot represents a SNP, a single nucleotide polymorphism, the higher it is, the more significantly it's associated with lupus. And these were very large studies. The first was a transancestral meta-GWAS that included 11,500 people with lupus compared to almost 16,000 people without lupus. And on the right, it's a GWAS that was primarily conducted in East Asian populations, again, including over 13,000 people with lupus and almost 195,000 people without lupus. And I'm showing these really to show that there have been multiple changes across the genome that have been implicated as associated with having lupus. And when you look at where those changes are located, a lot of them are near genes that encode proteins important in our immune system. So they've been implicated in encoding proteins important for clearing DNA debris from the circulation, important in encoding proteins like complement, important in the inflammatory cascade, and in T and B cell interactions. So all to say that a lot of this makes sense in linking genetic changes with what we know about immune dysregulation in lupus. Now, I'm going to sort of build on that a little bit and talk about how people have tried to use this knowledge and apply it to prediction. So these genome-wide association studies, as I mentioned, identify these alleles that are associated with developing lupus. People have tried to combine all these alleles to develop a score, an estimate of someone's genetic risk for developing lupus. They're called polygenic scores or genetic risk scores. And this was a study where they calculated polygenic risk scores for a range of different common polygenic diseases: coronary artery disease, type 2 diabetes, IBD, breast cancer. And they were able to see how well these risk scores predicted developing disease in an independent population. And for most of them, it was pretty good. Interestingly, they then compared how that score performed against a high risk allele. And they used familial hypercholesterolemia as their comparator. So familial hypercholesterolemia is a variant that confers a very high risk for coronary artery disease, a threefold increased risk compared to people who don't carry these variants. And these variants are rare. Less than 1% of the population carries these variants. What this study was able to demonstrate is that when they identified people who had very high polygenic risk scores for coronary artery disease, they had risks that were comparable to people who carried these rare variants for hypercholesterolemia. And so they were able to show that having an extremely high burden of these common variants would put people at risk comparable to people who carried the rare variants. So it's a really interesting phenomenon about a demonstration of how, you can learn from both studying polygenic forms of disease and monogenic forms of disease, and how they can shed some insight into both. So I think overall, what that study really nicely demonstrated was that polygenic risk scores can be powerful predictors for disease, that the effects can be comparable to monogenic disease, but it really does beg the question of whether these are clinically useful. The one thing to caution is that it can be really difficult to translate a population-based estimate, so what you're seeing from studying tens of thousands of people, to an individual person's risk. That can be very difficult because there are so many other factors that we aren't really able to capture in a lot of those other studies. The other consideration is where these polygenic risk scores were developed, what populations have contributed to them. One thing we know very well is genetics is systematically different depending on what region of the world your ancestors come from. We know, looking at someone's genetics, where their ancestors come from, because the genetic structure on people of African ancestry is very different to people from East Asian and European ancestry. And these are what some of the plots are showing here, is that if you were to compare African with East Asian populations, you're getting different correlations within the genome. And we also know, unfortunately, that the vast majority of genetic studies have been on European populations. This is from the same study that has shown over time how individuals included in genome-wide association studies have predominantly been European. So as of 2018, nearly 80% of people contributing to genome-wide association studies were of European ancestry, where only 16% of the global population is of European ancestry. This is a major problem that plagues a lot of genetic research, not just research for lupus. But I raise it today because I think it is particularly acute for diseases that we know do not affect these populations equally. We know that lupus is a disease that is more common among non-European populations. And so I think it's really important for us, when we're considering research and studying genetics, that our studies are as inclusive and representative of patients when we're doing this research as well. This is a map that came from a paper, a review in 2021, where they were estimating the prevalence of lupus globally. And as I was just showing, we know that there's a higher prevalence in people of African ancestry, yet it's really sorely underrepresented in genetic studies. And I think here it's interesting, they decided to shade the United States as a wide range, but I would also argue in Canada, we actually have a very wide range of prevalence depending on which populations you are focusing on. We looked at the epidemiology and ethnicity of the cohort at SickKids years ago. And I should mention, it's estimated about 20% of people with lupus are diagnosed in childhood or adolescence. And when we compared the ethnodemographic of our SickKids lupus cohort to the metropolitan Toronto at-risk population, we observed that there was a higher prevalence, relative prevalence, of patients of Asian, Black, South Asian, mixed ethnicity compared to the general population. And this, of course, is not unique to our clinic. This has been reported in multiple populations across the world. So what are we trying to do? I think lots of groups are working really hard to try to close the gap in knowledge. This was a paper published in 2021 where they were comparing the genetics of East Asian and European patients with lupus. And what you see in this far right is Miami plot. I guess it's supposed to be like a reflection of in the water or something. But anyway, a lot of the peaks between European GWAS and East Asian GWAS are similar, like you're seeing here in green, but there's also a lot of differences, a lot of genetic changes that seem to be only present or having stronger effect in one ancestral group compared to another. So I think work like this can be really important to highlight the similarities as well as the differences between ancestral populations. Also building on this utility of polygenic risk scores, this is work that came out of Boston using the Nurses' Health Study, which is a really fabulous longitudinal study of women within the United States and looking at the polygenic risk score in addition to other known risk factors for developing lupus. And what they're showing here in this ROC curve, where you're trying to predict with good sensitivity and specificity, so not getting too many false positives or false negatives, how they could best predict who would get lupus. Getting something up here in the top left is ideal. But what they were showing is when they used a model of a genetic risk score only in blue, their prediction was on par with family history, which maybe isn't all that surprising. But as they added things like lifestyle, they had the best predictive power when they put everything together in brown. So I think what it tells us is you're getting something different and getting additional information by adding genetics to other environmental predictors. So I think there is a potential when we kind of take it all together that maybe this can be a useful tool providing added information. Okay, the third question, this is a true or false. There is evidence that genetics influences age of lupus onset. True or false?
Waiting to see what the answers come in. We have a few trues.
I'm glad people are still awake. I haven't put it to sleep. Okay.
We got all trues so far.
Yes, the trues are right. That is true. There is evidence that there's a relationship. So let me share some of that. I think, firstly, why have people even considered this? Well, I want to share that there is evidence that when people are diagnosed in childhood and adolescence, that it tends to be more severe. And the reason why people have said this is because there tends to be a higher prevalence of lupus nephritis, lupus kidney disease, neuropsychiatric lupus, and hematologic manifestations when people are diagnosed at younger ages compared to older ages. And people have thought this might be a consequence of a higher genetic burden. And there have been a couple of studies that have shown an inverse association between the number of lupus risk alleles a person carries and their age of diagnosis. And here are two plots where they're showing just that. So on the left, you can see the older someone is, it seems that they have a lower number of lupus risk alleles. And here the axes are flipped, but similarly, they're saying that a younger age tends to have this higher burden of genetic risk. More about monogenic lupus. So again, this is that inverse association I showed you before, but this is in a subset of our patients that I just mentioned we had genotyped. So we calculated a polygenic risk score, and you can see that as someone is diagnosed at younger ages, these are bins of 5 years, the burden of lupus risk alleles tends to go up. But the section I'm not showing you is this section where you have the youngest patients, and granted they're a small number, diagnosed under 5 years of age, but they don't have the highest burden of these common risk alleles. So why are they getting lupus at such a young age? So this brings us to monogenic lupus. Monogenic lupus refers to, and again, please choose the phrase that best completes the sentence. A, lupus that is not influenced by genetics. B, a change in one gene that leads to lupus. C, changes in over 100 genes that increase the risk of lupus. Or D, a change in the gene related to mononucleosis or EBV infection that leads to lupus. Lots of people like this question.
Yeah, we're getting lots of answers, and they're all B. Oh, one C, several B's and one C.
Okay. So the B's have it, guys. Monogenic lupus usually refers to a change in one gene that leads to lupus. And really, this is not a new concept. In fact, the earliest recognized form of monogenic lupus was a complement deficiency, where people were not able to create enough complement, and so they weren't effectively clearing, mounting a typical immune response. But since the advent of next generation sequencing, when we're able to characterize nucleotides individually across the entire genome, more and more changes in these genes are being recognized, and they're involved in a range of immune functions. And so one project that we had conducted last year, it was published, was work done by a student in my group at the time, Melissa Mistal, who's pictured here. And what she did was she took data that we had created, sequencing 71 patients that we cared for in the lupus clinic, and looked specifically for rare variants in known monogenic lupus genes. We included 36 at the time. And she also calculated polygenic risk scores using common variants in those same patients. What she found is 13% carried a rare predicted damaging variant in a known gene for monogenic lupus. And here are the genes listed here. And I should mention, this was a very selective population. We had sequenced patients who we suspected were more likely to harbor these rare variants. So we had selected patients who were younger than the median age of diagnosis, so less than 11 years of age. They were born to parents who were consanguineous, so they were second degree or closer, and patients who had a first degree family member who was affected. Then Melissa also looked at the relationship between these rare variants and the common variant PRS to see if there was any relationship. In short, she didn't find a relationship. So overall, what Melissa found is up to 13% of patients in this selected population carried rare monogenic lupus predicted damaging variants. The patients that did carry these variants tended to be younger at diagnosis, and we didn't observe an association between common variant burden and rare variant burden. We suspect this may have been due to the very small sample size we had. So I'm going to delve into one of those genes a little bit more, just to make a second point. So DNase 1L3 is one of these enzymes that has a really important role in our bodies. Our cells are constantly turning over. They're dying and regenerating. And as a consequence, we have DNA fragments circulating in our bodies. DNase 1L3 is one of these enzymes whose job it is to sort of gobble up and break down the circulating DNA. And that means we're not going to have persistent DNA fragments in our circulation. When we do have persistent DNA fragments, they can interact with activated B cells, so a type of white blood cell that can then create autoantibodies or self-antibodies against this DNA. And then that can lead to clinical symptoms of disease. So this is one of the demonstrated mechanisms by which variants in DNase 1L3 can lead to lupus or lupus-like disease. And I want to introduce you to two sisters that we met years ago, and we have their consent to share their story. They were of Turkish ancestry, born to parents who were first cousins. The first sister was diagnosed with lupus at 2 years of age. Her disease was manifested by joint pain and swelling. She had low complement levels. She had a very high titer antinuclear antibody. She also had recurrent fevers, swelling of her lips and her face, an urticaria or hive-like rash, and red eyes. Her sister was diagnosed at 5 years of age. Her disease was quite different. She did have fever and arthritis, but she also had bleeding and bruising and was diagnosed with lupus nephritis or kidney disease on a biopsy. Unfortunately, when we met her, she was dialysis dependent. Seeing these girls, knowing this story, knowing what region of the world they were from, we suspected that they carried variants in DNase 1L3. And when they were genotyped, sure enough, they did. They were both homozygous, or they carried 2 copies of a deletion in DNase 1L3 that was predicted to result in a premature shortening of DNase 1L3 enzyme, which then led to its degradation. So it basically meant that they were DNase 1L3 deficient. When we looked in the literature, this same change has been reported in 3 other families. And also there was another series reported in a couple of other families who had other changes in the same gene, but all had features of lupus with varying overlap with the symptoms that our patients had. What was interesting about this particular study that I'm sharing is that when you compare these patients who have lupus as a consequence of DNase 1L3 variants, their interferon signature, interferon is one of these cytokines that's been shown to have play a very strong role in lupus. But compared to other interferonopathies, the level of interferon stimulated gene expression is variable in DNase 1L3 patients, meaning it's not persistently expressed, which also gives some hope that these patients can have disease control over therapy. And that's one thing I'd like to hopefully convey, is that these girls both managed to have good disease control when we understood the disease a little bit better. Interestingly, there was a small study that looked at 20 patients with lupus and 26 people without lupus, and they compared their DNase 1L3 quantity and also activity. And the one thing I want to emphasize is these people did not carry rare variants for DNase 1L3. These were people who had presumably a polygenic form of lupus, but they too demonstrated lower levels of DNase 1L3 and DNase 1L3 activity compared to non-lupus controls. Going further, since then, there was another study that demonstrated that people with lupus kidney involvement had even lower levels of DNase 1L3 activity, and also demonstrated higher levels of anti-DNase 1L3 antibodies. So this was a really interesting piece of work that showed a mechanism by which someone could acquire a DNase 1L3 deficiency, that people could develop antibodies that then could neutralize or make DNase 1L3 inactive. And that could potentially lead to all the downstream consequences that we can see in people who have monogenic forms of lupus. So what I hope to demonstrate with this specific example is how it's really important to study the entire range of lupus from a genetic standpoint and from a clinical phenotype standpoint, because there's a lot that we can gain from studying monogenic forms of disease and polygenic forms of disease and how they can inform one another. So I think with that, I had wanted to talk about the female predominance, but I wonder, Lisa, if maybe I'm going a little over. Do you think we should maybe pause, or what would people like to do? Because I do want to leave time for questions.
Yeah, we don't currently have any questions in the chat. I have some that I would like to ask as we go forward. So yeah, do you mind if we pause and I ask a few questions and then
Of
we can?
course, absolutely. Maybe
Okay,
I'll stop sharing and then we can
that would be great.
get
Yeah.
one another. Yeah,
So again, if anybody has questions, please put them in the Q&A. I also have the chat open so I can see them there, but we'll do with that. So there are a few things that I was wondering about. I was wondering, early on you spoke about 10 times concordance for MZ twins. I forgot what the MZ
yeah,
stands
yeah.
for.
Monozygotic,
Thank you.
identical
Yes.
twins.
So do you know why it is more prevalent for that form of twins? Has that been actually identified?
So I think I was using it as an example for why we think genetics plays such an important role, because we know that in people who are monozygotic, genetically they are identical. They're because they are basically the product of a, a gamete. I'm sorry, I'm trying to think of like another term for it, but they're genetically identical compared to dizygotic twins, who, even though they might share the same womb, genetically they are not identical. They're the consequence of two separate eggs and two separate sperm coming together. And so that's why genetically they're actually like any other set of siblings, but they just happen to share a womb.
That is an amazing way to explain it. Thank you. I'm really happy that there are those twins out there that have this for the study. I'm sorry that they do, but that, yes, to get you that kind of good data. It was surprising to me, I think, that there was 66% heterability for this. Like, I knew it was prevalent, but that was quite large. And I think you did a really great job of explaining to us the variety of genes that could be affected, or the alleles in the genes and that. So again, contributes to just how complex this disease is for so many of us.
Yeah.
What I was wondering was, you had said there is you're identifying a high burden of variances comparable to having a rare component. And I was just wondering, could you just explain that just a little bit further?
Sure.
Make sure that we've kind of grasped that.
Yeah, I mean, I think the idea is it's it can be really, really hard to predict for an individual. That's just one thing I'd like to say up front. I think there are a lot of genotyping companies that try to give people like a formula of how they can predict their future, but that can be really challenging for these individual common variants that have an estimated effect that's really contingent. Well, right. They may not know how many people are smoking, how much they're smoking. And we know that behaviors like that play a really big role in a person's individual risk. So what I was saying is, like, this was sort of an extreme example where they were able to look at a subset, about 20% of the people who had a very high burden of carrying many of the common risk variants for cardiovascular disease. And in that small subset of the population, their risk was comparable to someone who carried a single genetic variant of strong effect. So that was what I was hoping. I'm sorry, it was a little convoluted.
No, that's great. Thank you just for the recap of that. That is really helpful. Um, You also talked about those genetic studies being so representative of people with European ancestry. Is there, and I saw where they're trying to increase this. Is there an ongoing effort in the medical community to uh, represent the ancestry better overall, or has it just been identified this is a problem? Is there an active push for the change?
Absolutely, 1,000% there is. I think that's happening in an international level, a national level, and local levels. So I know there is a massive effort within Canada to try to engage and include people of a range of ancestral backgrounds. In particular, not only underrepresented, but marginalized communities like First Nations Aboriginal populations, who we know have a significant burden of disease, but who have traditionally been excluded. So I think there's a massive effort to include and partner with those communities to make sure that they're represented. I know in the US, there is a massive undertaking for the All of Us study where they're trying to sequence about 1 million people and also being inclusive and representative. And of course, there's more efforts across the globe to try to do this as well. I think there's still a long way to go, but I can tell you that we had specifically selected genotyping platforms that were designed to include genetic changes for all ancestral groups. And those are the platforms that we used for our studies that I was sharing today, because we know that our populations with lupus are quite ancestrally diverse.
Okay, that's great. We have a couple of questions from other people, which are really good connectors here. So one is, if there's a 66% chance of passing SLE to offspring, does that mean it increases over time?
That's an interesting question. I just want to be clear, though. I wasn't suggesting there's a 66% chance. That's not what heritability means. Heritability is saying, okay, we have a risk of developing lupus. What proportion of that is due to genetics and what proportion of that is due to other things? But again, that can't be translated into a person's individual risk. So just, I hope that clears that
Yep, I think
up a
so.
little
Yeah.
bit more.
Yeah.
So there isn't this sort of generational intensity of effect. I think there are other examples in medicine where that actually does happen, where you can, they call it penetrance, where you can have changes in the genome that become more severe with generations, but that's not a phenomenon that's been commonly observed in some of those monogenic traits, and it can be kind of difficult to quantify for some of the modest effect variants. I think we do know, again, this sort of just speaks to the heritability, that people who have a first-degree relative who's affected with lupus, their individual risk is higher, but specifically how much higher is very, very difficult to quantify.
Good to know. Yes, that's yeah, knowing that there's that first degree connection helps, but yeah, not knowing the percentage of it is perhaps in time that will come.
Yes,
No.
yes, we're trying.
Yeah. Um, Someone has asked, what is considered a pediatric diagnosis of lupus? I understand it can take a long time to get an accurate diagnosis. So what's that age range?
Yeah, it's a really, really great question, because I think as you're alluding to, there can be delays in when a person is actually diagnosed. So different people define it differently. It's probably a consequence of medical systems as opposed to true biology. I'd say usually in North America, it's under the age of 18. Some cohorts in the UK will use 21 or even 16. So I try not to get too fixated on that definition. I think, we're talking about a continuum. And I think the other challenge is sometimes people will have first symptoms years before they actually evolve and someone makes the connection that, oh, this is all the same process. So I think that also makes it really difficult to know, okay, at what time did lupus begin? Because as we know, it's a great mimicker. It may come on as some low counts, some joint aches, a rash, and it takes years before someone evolves and develops more symptoms that they're able to all put together and realize, okay, lupus is the unifying diagnosis here. That
Yeah,
was a
so
very wishy-washy answer. I don't know
well,
if that's
no, but I think you've hit it, is that like there could actually be a higher rate of pediatric onset lupus if it was diagnosed at earlier ages, and simply because of the way the disease is and the system of our healthcare and the way it presents, it becomes adult onset determined, even though you've had symptoms since being younger.
Yeah, absolutely.
Another question is, following from your question about representation, is there partnerships happening with universities, organizations to increase and understand the differences? For example, HBCUs in the USA or University of West Indies in the Caribbean?
Yeah, that's a great question. I'm not aware about large initiatives. I can say personally, at least at SickKids, there is, from a clinical standpoint, there's a partnership between SickKids and children's hospitals in the Caribbean. And so I've personally been involved in some of those consultations, more on an individual case-by-case level. You know, if they've suspected a monogenic form of disease, I've been able to help with facilitating some of the genotyping and sequencing. But beyond that, I'm not personally aware of anything. Doesn't mean it's not happening. At the end of the day, unfortunately, it really comes down to funding and resources. And I think oftentimes that's the biggest hurdle. I know there was a specific initiative that NIH in the US had supported to complete sequencing of people in Africa, but even that was a small fraction of the African population. We know the continent of Africa is probably the most genetically diverse of any region in the world. And so even that undertaking is massive. So I think the approach has been with incremental gains, and I think there's a lot that has been done, but still even more to go.
Yeah, and one of the things we do at Lupus Ontario is we try to shine a light on any studies that are available that patients can participate in. And we highly encourage you, if you're listening to this, to participate in those kinds of studies so that we get all this great data, that these doctors are not just gathering the data to just let it sit on a shelf. They, as you can see, Dr. Aki, they're using this like right in their practices and to teach others. So anything that they gather from us as lupus patients can help the medical progress moving forward. I know we're just about out of time. I wouldn't mind asking one more question, and then if you have anything else to wrap up, I know we won't get to your the next few slides you were going to talk about, but so when you were talking about the DNA SE1LE, I think I've said it wrong, but
DNA is 1L3, yeah.
it's 1L3. Does, to put it in very simple terms,
Yeah.
does anybody who has a deficiency in that have SLE? Or not, we don't know for sure.
So, okay. So I think there's a couple things I kind of glossed over. I made it sort of a neat and tidy story, but it's not very neat and tidy. Because I think the one thing that I'm sure everyone could appreciate was even these sisters who have the identical genetic change in the same gene, their disease looked dramatically different. So why is that? Is that an issue of variable penetrance or variable effect of the same variant? Is it because that variant in and of itself is not acting, that there's also the influence of all their other genetics that is not identical? Does it have to do with the relative contribution of other exposures? Maybe they didn't see the same infections at the same times in their lives. These are the things that I think we still don't understand. And so that's why I can't say 100% that that's the case, but I will say that what makes it very likely that that this is causal for lupus or lupus-like disease. And again, I didn't talk very much about lupus-like because you're like, well, what does that even mean? I mean, we all know that there is no single test for lupus, right? It's whether you have it or not has to do with the impression, and whether, there's a better explanation for your disease. So at the end of the day, really, what does it matter? I think it's sort of how we treat a person and how we try to get them better and feeling better. And that's why sometimes these labels can be useful, because they can help us direct us to our therapeutic options. But frankly, these two sisters, again, who had identical genetic changes, the disease was really different. And so we treated them very differently, because we shouldn't have expected them to respond to the same thing, because they were so very different. So I think, yes, as far as we know, this genetic change is linked with a very, very high risk of getting lupus at a very, very young age. And that is something that has been consistently shown in those other patients and families that have been reported in the literature. But what I also wanted to point out is that directed the community to investigate DNase 1L3 more broadly. And that's how people were able to discover, oh, maybe there's an acquired version of this, and it's playing a role in another group of people who develop lupus later in life. So that's
It
it.
it is so fascinating, at least it is to me, and I'm sure it is to other people listening, because our bodies are so complex and there's so much to study and learn. And then when we have a systemic disease that is also complex, I know it provides endless amount of information to be gathered. Um, thank you so much for your time, Dr. Hiraki. Thank you to everybody who's attended tonight. Did you have any final words that you wanted to say about tonight, or
I
are we
just
good?
really appreciate everyone taking the time out of your evening, because I know, these evenings can be precious. And so thank you very, very much for spending it with us. And I certainly hope if people have burning questions, you're welcome to reach out to me. So thank you for having me. I
Well,
appreciate
thank you so much for attending. And yes, like Dr. Hiraki said, if you have questions, you can reach out to her, or you can reach out specifically to us at Lupus Ontario about anything related to this. We'll have most of this recording available. So those that are listening on YouTube now, you'll see a section was cut out, but there's really great information. So, and perhaps we can do something about the prevalence of women at another time.
Absolutely. Thanks,
All right.
everyone.